So in the last part of our unit, we're going to talk about biotechnology, which is basically applying our knowledge of genetics and DNA and using it to figure stuff out and to potentially modify genes. So we'll start by talking about DNA tools, which is things like when we get a DNA match on TV, what exactly that entails. And then we will talk about genetic engineering. Something important to note is that this changes a lot. So you'll notice that this is the second video I've made. I'm actually going to try to save a little bit of time by splicing some older sections in with the new video. So since it's such a rapidly evolving field, focus less on the little minutiae and the tiny details and more about the general themes throughout, like the idea that we're going to be able to use base pairing to copy genes. Make sure, as always, you come see me if you have questions. So to start out with, we'll talk about a very important DNA tool called DNA fingerprinting. If you are familiar with forensics at all, or you ever seen a TV show, you know that we leave behind fingerprints when we touch things, and those fingerprints are unique to each person. So if we have a fingerprint, we can match it to the source. DNA works very similarly. Everybody's DNA is unique, save identical twins. And because of that, we can compare DNA samples. So if somebody leaves behind a tissue sample, and because all of our cells have the exact same complement of DNA, it can be a blood cell, a skin cell, a spit cell, whatever it is, if we have a tissue sample from somebody, we can compare that tissue sample and match it to another sample from their body. Additionally, because relatives have very similar DNA, we can also compare familiar relationships. So if you've ever heard of 23andMe or Ancestry.com, where people can say that they learn about their ancestry, they're looking for specific sequences in DNA. Sometimes on those programs you can even find you had a long-lost cousin or, depending upon your family situation, a long-lost sibling or something, because family members' DNA should be very, very similar to each other. The main method we do this is a strategy called gel electrophoresis. So, quick story. When I was in high school, there was the O.J. Simpson murder trial, and I remember I was homesick from school one day, and my family didn't have cable, and it was before the internet. Well, before most people had internet. And so watching TV and daytime television sucked. And so all I had on was news. And at that time, all of the news was the O.J. Simpson trial. And I remember hearing person after person talk about the DNA evidence. And there was this big, huge concern. Would the jury be able to understand it and follow it and believe it? Because, of course, a lot of times in science, if you don't understand something, you don't necessarily believe it, which is horrible and shouldn't be the case. But in any event, there was a huge debate about whether there was just too much DNA evidence. Now, of course, thanks to TV shows and movies, if someone says there's a DNA match, almost everybody accepts that as like, okay, yeah, that's a real thing. But it wasn't always the case. And as an adult, one of the things that biology will impact you, obviously there's a lot, but one of the ones is you're likely to serve on a jury in which DNA evidence might become important. Just for Christmas, I got an ancestry kit. You probably will have experience to get that pretty soon, or even at your doctor, test for certain genes. So this is all pretty important. So, like I said, the main way to do it is using a strategy known as electrophoresis. Electrophoresis uses an enzyme called a restriction enzyme. A restriction enzyme is an enzyme that can cut the DNA, and when it cuts the DNA, it does so at specific sequences. So there's different kinds of enzymes. The one in the diagram is EcoRI, and it cuts it at that pattern where one side is GAATTC, and then the complementary, of course, would be CTTAAG. Whenever it can scan through the sequence of DNA, and whenever it sees that, it can cut it. And when it cuts it, it leaves behind what we call a sticky end. In other words, it cuts it on a zigzag, so that on one side, TTAA is sticking out, and on the other side is AATT. And because those are complementary to each other, those two things, remember, they bond to each other by hydrogen bonds, and so they're looking to form another hydrogen bond. So that leaves a sticky end or a charged end. Because it leaves pieces of DNA that have charged and it's cutting at a particular sequence, when it cuts an individual's DNA, it cuts them into different pieces. And so because my DNA sequences aren't the same as yours, the pieces where it sees that sequence in my DNA will be cut into different pieces than where it sees it in yours. So what happens is we put the DNA into a gel, and at the one side of the gel there's basically little holes that we call wells. And so the DNA will be put into a gel here. They'll run a charge through it, and because the DNA has negative charge, when it's cut by the restriction enzyme, it will be pulled towards the positive side. And when it does that, the heavier pieces, the larger pieces, won't have as much of an easy time moving through the gel as the smaller pieces. And so the heavier pieces or the larger pieces will stay closer to the well. Some won't even make it out of the well, and the smaller pieces will be pulled all the way through to the other side. And in that way, it will create a band that's unique to each person. Back in the day, and even still a lot of times today, it looks something like this. They use a UV light to see it with special dyes. Now, of course, they have computer-enhanced methods of doing this, but the old school method, which is still tried and true, is to put it in and to analyze it using a picture to take a look at the actual dye images. So once the DNA has run through the gel, each person's DNA, being cut up in different pieces, will create a banded pattern. And if the banded pattern is from the same source, it will be the same band. So in this case, the purple band and the black band are exact match, and so that can be conclusively said that that is from the same person. Whereas this band, there might be some lines that are kind of similar, but the purple bands and the black bands definitely aren't matched. In addition to matching a person's DNA from their own source, like a spit sample to a blood sample, we can also, like I said, match family members. And of course, the most important match that we can make is from parents to child. So because half of an individual's DNA comes from their mom and half of the individual's DNA, approximately half, comes from their dad, each of the bands created in a child's DNA should come from either their mom's segments of DNA that they got or their dad's segments of DNA. So as we look at the gel pattern from a kid, every line in the pattern should correspond to one of its parents. And so because of this, if somebody doesn't know who one of their parents are, or both, they can do paternity testing, or depending upon the situation, maternity testing, and find out family members. Of course, siblings should have a lot in common, but because of crossing over and because of independent assortment of chromosomes, they won't all necessarily match a sibling. Obviously, like I said, this is incredibly helpful in criminal cases. Like we have here, we would have a victim and perhaps a blood specimen that would be found at a crime scene that doesn't match the victim because, of course, we've got a band here and the victim doesn't have a band. So we know that this is from somebody else who was at the crime scene. Not necessarily the perpetrator, but certainly somebody who was there to leave blood or spit or whatever the sample is, and so we can match it to an individual. And of course, if it's an exact match, it would have all of the bands in common. So suspects 2 and 3 can be eliminated as the source of that match. The gel on the right would be a photograph of a real gel with 2 potential fathers. And so this child here, all of their bands would have to match one of the 2 fathers. And of course, we can just tell from the first couple of bands here that this one matches father number 1, but not father number 2. So the individual who's labeled as number 1 would be the parent. Nowadays, it looks something like this because, of course, we're not taking a photograph of a gel, but rather a computer is analyzing it. This is incredibly accurate. So it's so accurate that it can be highly reliable in court. Of course, there's always matters of, to be taken into account, like just because a person had blood there doesn't mean that they were there when the crime scene was, when the crime was perpetrated. But it's definitely an important tool that, as a potential jury member, you want to make sure that you understand this. Going back to O.J. Simpson, he was acquitted, even though we have this DNA, and even though it's a super old example, here's one of the victim's blood, his ex-wife, and here's blood that was found in his car, maybe, I don't remember. He had a white Ford Bronco, and I don't know if it was a glove or whatnot, but nonetheless, it was found associated with the crime scene, and it was a match, but the jury didn't put enough stock in that. Before moving on, let's take a few moments to review DNA. We talked about electrophoresis and DNA fingerprinting in class. There are some other things like PCR and DNA sequencing that we'll talk about here. So you might have seen on TV that sometimes a very tiny DNA sample, like a speck of blood, has been left behind at a crime scene. PCR is a way of making sure that there is enough blood to test. So PCR stands for polymerase chain reaction, and it's basically a way of taking a small sample of DNA and increasing the quantity so that there can be lots of DNA. The steps of PCR are actually pretty straightforward. The first step is to heat up the DNA. When we heat up a molecule, it moves faster, so that DNA starts to vibrate more and more. Well, remember, hydrogen bonds are the only thing holding together the two strands, and although they're strong in numbers, they're not strong enough to withstand really, really fast motion. So those hydrogen bonds break. When the bonds break, then we cool down the temperature, and the strands are now separate, and we can take a primer, which remember is just something that starts replication. We can take a primer, the primer attaches on, and DNA polymerase can build a new strand by complementary base pairing, just like we saw in replication. Now we went from having one strand to those two separate strands, each having a new side, just like in replication. When we repeat the process, we go from having one strand to two strands, and now those two strands become four strands, and so forth. Here's a video that shows it real nicely. Let's take a minute and review. The first one is DNA sequencing. And DNA sequencing is really just a way of determining the order of the nucleotides. So what is the AACGGTC code in a particular gene or molecule? We do that by labeling the DNA, then making pieces that we can cut. And the pieces of different size we're able to pair up and see the entire length. I'll show you that in a second. But let's talk briefly about why we would do that. So we would do it to find the order within a particular sequence of DNA, like within a gene. We might do that to help predict the function of the gene, to help determine what the amino acids will be. We can compare genes of similar sequences to different organisms, like we did with insulin and cow DNA. We can identify where there are mutations and what the effect of those mutations would be, and a great number of other things. To sequence the DNA, what we do is we take an unknown sequence, like the one on the left. We don't actually know what that sequence is, but in this picture we do. And we would have 4 different solutions put in. Each solution would have a base that is labeled. In other words, we would have bases that we know what they are. And so in this solution, there would be regular nucleotides and then labeled Gs. And those G nucleotides would each be modified in a way that when they're put down, another base can't attach to it. And so what that happens is every time we put this G down, we put one here and we put one here, we'd have strands of different lengths. Same thing with the A and the T and the C. But then we would put the pieces through electrophoresis, and what that would do is it would create bands of different lengths. And so we would know that the bands ended in each of these letters. So the first band is G, G, and then T, T, and the next biggest pieces, and then T, and then A, and so forth. And we can actually see the sequence of DNA. This sounds a lot more complicated. It's actually kind of straightforward, and it's a great way of actually finding the sequence of a segment of DNA. There were a lot of things that were discovered by the Human Genome Project. The first is just how big DNA is. It's over 3 billion nucleotides long, and that there are over 23,000 genes. We still don't know what they all do. That's where things like RNAi come in handy. It's important to note that the 23,000 genes that we have only represent a small portion of our DNA. There's a lot of sections between the genes that are non-coding. The coding section, in other words, the ones that actually get turned into proteins, are pretty similar because, of course, all humans have pretty much the same recipe for saliva. But there's a lot of differences in the non-coding sections, and it's those non-coding sections that we use to do a lot of our identification of DNA fingerprinting. Let's take a minute and review one last time.